All right, with chapter 13 coming to a close, we have ourselves our project, and this project kind of builds you up one step at a time. And as we can see on this post right here, this assignment, it says Chapter 13 Project, Step 1. And Step 1 is investigating a new technology in genetics, and it's called microarray. And microarray is a really interesting, very powerful method and technology for studying DNA and better understanding what may be the genetic causes of a particular disease or someone's phenotype, appearance, their behavior. It's a way to, generally speaking, look at someone's entire genome, all the DNA in their body, all the genes within their DNA, and studying it in a very quick manner and getting a lot of good information or results back from it to fully understand what's happening. Quickly learning what genes are active, what genes are not active in someone, particularly a sick person. Without saying more, it's your opportunity to study microarray. As you can see, it's titled right here, microarray. And we're doing something a little different this time. We have a Google Slide document, and this Google Slide document allows you to, as it suggests, summarize information. What's different, though, is instead of reading some information and then reading something and then writing summaries about it, you're going to actually listen. And so here's the audio file 1, and that's the initial information you should listen to. After listening to this audio file, you should be able to write up to 10 important ideas or details about microarray. And you'll write those details. So while you listen to this, you should stop, pause, replay, do whatever you need to with the audio file. But pause it as you need so that you can take time to go into this document. And just as you see here, writing facts along the way. Hopefully better than that. So you'll listen to the audio file, and as you get important ideas, or what you think are important ideas about microarray from the audio file 1, you'll record them here. You'll end up with a list of up to 10 ideas or important details about what microarray is, what it does, how they do it, etc. Once you've done that part, then you're going to move on to the audio file number 2, and you simply repeat the same thing. You click on audio file 2, you play it when you hear details about the process of microarray, what it does, why, how, all those important what, why, when, etc. ideas. When you hear those, that's when you stop, pause the tape, or replay it so you can hear it again, and then you take that information and you type that information in right below here for microarray 2. Write down as you go, right? So you type it as you go down through here. Easy enough. Once you get there, you're pretty much done with the 2 audio tapes. The next thing, once you get enough details from these 2 audio tapes that explain what microarray is, how it works, what it's used for, why it's useful, when you get those important ideas and details about the process or that technology, you then are done with the audio files, and then you proceed to the last step. Let's look at this. So the third page in this pre-project activity is it says right here, combine notes in a clear summary. And then second of all, it says, as you combine your notes in a clear summary, you are to also add all images from slide 1. We're going to correct that. It should say slide 2. So you add all images from slide 2 to provide visual support to your ideas. This is something new too. So what you're going to do is you're going to write out a summary of what you understand microarray to be based on what you heard from those audio files. As you do that, you're supposed to go to page 2 here and use these images. So you'd copy, and you'd take them and you'd paste it on in here. So for example, and also it's a hint, the microarray process in general is going to utilize genes that are active, inactive. Here we have DNA where something's happening to the nucleotides. We could zoom in a little better. Let's see what it looks like. Looks like radiation is changing the DNA. So if you remember what changing DNA is, you got yourself a mutation. We have right here a picture of that process. What's interesting is you can see here you have a normal sample of DNA strands or genes, or potentially mRNA, but some form of nucleotides. And then it says you have disease samples. So you have strands of some kind of nucleotide, whether it be mRNA or DNA, you'll find out, that are associated with disease. So normal and disease. So you get it. So this discusses a comparison between normal nucleotide samples and disease nucleotide samples. And then right here, we have, what's interesting is this whole platform of different colors, and each color represents something different. So we can zoom in on it. And it says if the circle is gray, it means that area contains genes not active in either a normal or diseased person. So if you're studying people and you find out that when you're comparing a sick person and a non-sick person, and neither gene is active, that probably means, because it's the same in both people, probably isn't something you got to worry about. Here it says a gene is active in both normal and diseased samples. Here, this is probably telling you also that these possible yellow genes aren't involved because it's the same in both people. So really what you're trying to do, if you haven't guessed it already, is what's unique about a sick person? You know, what genes are different in a sick person compared to the healthy person? And this should indicate to you what gene might be causing the disease in someone. And this is how medical people study this nowadays with genetic technology. Look at the green, means the gene is active in normal people, and then, which is an important detail, what genes are active in normal people? And in here it says genes active in disease only. So what are the genes that are active only in the diseased person? So when you know which genes are active in normal, which genes are active in unhealthy people, and which are the same with both people, it helps you narrow down the possible genes involved. As you summarize the information, these pictures are going to be inserted. Finally, we have here DNA, and then the strand of DNA is being converted into an mRNA strand because that gene is active and it intends to make a protein, right? But before it can make a protein, DNA has to convert nucleotides into mRNA, and that mRNA strand, of course, then goes to make a protein. And the proteins are parts of your body, right? And your proteins keep you alive, they do everything. So your whole body is proteins, everything, right? Alright, so case in point, these pictures are used on this last part here. So just to give an example, we have a gene here. Yes, that is correct. It is a non-active, or it's a gene that's turned off. So let's say I was typing here and say let's zoom in a little better. Let's make this a little simpler. There we go. We'll just increase the font. Some people whom are sick have certain genes turned off. And then what I would do is I'd actually take this picture, and this is a way for you to show that you know what a gene looks like when it's turned off, but it's also a visual support in your writing. And you can move on. However, and this isn't what you should write, it's just simply an example. However, some sick people also have genes that are turned on. You can see already the picture's a little too big because it's cutting off the bottom. But this is exactly what you do. You copy, paste, you drop, And now I'm going to take the turned on version. Here we go. Copy it. Drop it over. And paste it in there. And presto. You got yourself an image showing it active. So you're not only supporting your understanding, so you're summarizing, okay, and showing your understanding of what microarray is, but at the same time you're adding pictures just to support what you're stating and to give not only a written understanding, but a visual understanding of the whole process. Alright. Without giving you more hints, I want you to have a chance to do well on this on your own. I want to wish you the best of luck. And remember, it's whatever ideas you think are important to explaining what microarray is, what microarray does, why it's useful, the steps and the procedures to effectively learn more about the genes in sick and non-sick people. As you write your summary, based on your observations, all 20 of them, and remember, as you write this summary, it should not just be your 10 observations connected together. If you were to take all 10 of these, and combine them, that's a tremendous amount of information. More information than you want to have to type up here. It also is pointless because you're just copying the same thing here and here. Your challenge as a writer and as a scientist is to take the 20 roughly, or the roughly 20, as close to 20 observations as you have, and connect these ideas in clear ways that are also new original ideas. So if this sentence said, genes in individuals can be inactive or turned off, and then here you put scientists that have determined that certain genes can cause disease. So you have these two sentences, right? Let's pretend that these were from the recording. Rather than just take these two sentences and type them out one right after the other, which is rather lazy, and it takes up way too many words. It's too wordy, and there's no point if you're trying to take new ideas and shorten them, right? So what we want to do is take those two ideas, and let's see them again. It says gene in individuals can be inactive or turned off. It says scientists have determined that certain genes can cause disease. What we could do in an original sentence here is say, and this is just combining the first two observations, just to demonstrate, you could write, scientists are studying which genes are turned on and which are turned off in sick and healthy people. There you go. And you could just take these pictures here, right? Turned on, turned off, bam. I just used two pictures, and I just created an original single sentence, used fewer words to capture the ideas in two whole sentences. There you go. That's how you summarize, and that's what I expect you to do here. But as you proceed through your process, you need to make sure to use every single picture here. If you find you're losing space, so as you type this and you realize you need more space, just simply add a new slide and then continue on. You do that by inserting a text box, throw it on in there. You can adjust your font as much as you want, and then continue to copy and paste as you need. Alright, good luck and get it done. Again, this is just the first step of many, so this whole project will be made up of up to 3 steps. One which will be a real lab where you'll be investigating using chemicals and studying the genetics of 2 individuals. Alright, good luck.